Every second, your body needs oxygen to stay alive. You breathe air into your lungs, and your blood carries this oxygen to your organs. But there is a huge problem. Liquid blood without cells is called plasma, and plasma cannot hold much oxygen. Let's look at the exact numbers. Plasma only holds 0.003 milliliters of oxygen for every 1 millimeter of mercury of oxygen pressure in 1 deciliter of blood. In your normal arteries, the oxygen pressure is 100 millimeters of mercury. This means the plasma only carries 0.3 milliliters of oxygen per deciliter. This is a tiny amount. It is not enough to keep you alive. To fix this problem, humans evolved a special protein called hemoglobin. Hemoglobin increases how much oxygen your blood can carry by 70 times. Hemoglobin is a tetramer. This means it is made of 4 smaller protein pieces called subunits. In a normal adult, this is called hemoglobin A. It has 2 alpha chains and 2 beta chains. Inside every single chain, there is a special ring called a heme moiety. Right at the center of this ring, there is one single atom of iron. The iron must stay in a special form called the ferrous state, written as Fe2+. When oxygen binds to iron, it is a clean and safe process called oxygenation. It is not oxidation, which means the iron does not rust or change permanently. It just holds the oxygen safely and can let it go later. Hemoglobin changes its shape to do its job. It uses something called cooperative binding, which means the parts help each other. It has 2 shapes. When hemoglobin has no oxygen, the 4 parts are locked together tightly by ionic bonds and salt bridges. This is the tense state. Because the pockets are small, hemoglobin has a low affinity, which means a low desire for oxygen. When blood goes into your lungs, the oxygen pressure gets high. One oxygen molecule forces its way into a pocket and binds to the iron. This breaks the salt bridges, and the hemoglobin now turns to its relaxed state. This causes a 500-fold spike, meaning it wants oxygen 500 times more than before. Because of this shape change, the graph of hemoglobin is an S-shaped curve. At low oxygen pressures, the curve is flat because the molecule is stuck in the stubborn state, but once the first oxygen breaks in, the curve shoots up fast because hemoglobin turns to its relaxed state. Let's look at the exact math of blood in real life. 1 gram of hemoglobin can hold exactly 1.34 milliliters of oxygen when it is 100% full. A normal person has 15 grams of hemoglobin in every deciliter of blood. So when blood is completely full, it carries 20 mils of oxygen per deciliter. Let's compare it to 2 types of blood in your body. Arterial blood, almost 100% full, so it can carry 20 milliliters of oxygen. Venous blood is only 75% full and can carry 15 milliliters of oxygen. When you subtract 15 from 20, you get 5 milliliters of oxygen. This is the amount of oxygen being used per 100 ml of blood. And when you multiply it by the cardiac output, you get 250 milliliters of pure oxygen, which is the amount of oxygen being utilized by your body every single minute. Hemoglobin reacts to its environment. We measure this using the P50 value. This is the exact oxygen pressure where hemoglobin is exactly 50% full, and it is at 27 millimeters of mercury. When you exercise, your tissues get hot and they make carbon dioxide and acid. This moves the graph line to the right, which helps hemoglobin drop off oxygen faster. There are 3 things which cause this. The Bohr Effect. When tissues make acid, the pH drops, and this acid decreases the affinity of hemoglobin for oxygen. Second is temperature. High heat from exercise or a fever breaks the bond between oxygen and heme, accelerating the release of oxygen from hemoglobin. Third is 2,3-DPG or 2,3-diphosphoglycerate. It is a highly charged chemical made during the respiration pathway. Once the molecule of DPG binds to the beta chains of hemoglobin, it decreases its affinity for oxygen. The exact opposite happens for a leftward shift of the oxygen-hemoglobin dissociation curve. An alkaline pH, and decrease in temperature, and a decrease in 2,3-DPG cause the shift of the curve to the left side. Here is an important note about banked blood. If your body is starving for oxygen or has low blood cells, your body makes more 2,3-DPG to get more oxygen. But inside stored medical banked blood, 2,3-DPG drops over time. If a sick patient gets old banked blood, the hemoglobin holds onto the oxygen too tightly and cannot release it to the tissues. Now let's talk about myoglobin. Your muscles have a special oxygen protein called myoglobin. It is a monomer, meaning it has only one part and one binding site. It does not have cooperative binding, so its graph is a simple curve called a rectangular hyperbola. It sits far to the left of hemoglobin because it has a very high affinity of oxygen. It steals oxygen from hemoglobin and stores it. It will not release its oxygen until the pressure drops below 20 millimeters of mercury during heavy exercise. Now let's talk about waste. Carbon dioxide. It dissolves in water 20 times better than oxygen. When CO2 leaves your cell and enters blood, it has 3 ways of traveling through the blood. It can travel dissolved in the blood, and 5% of CO2 does it this way. 5% can travel bound to hemoglobin. This is called carbohemoglobin compound. And the remaining 90% enters into red blood cells. Here it combines with water to form carbonic acid, which splits into bicarbonate ion and H plus ions. The H plus ions then leave the cell and enter into the blood, and in exchange chloride ions enter into the red blood cells. This is called the chloride shift. There is another effect called the Haldane effect, which simply means that empty hemoglobin has a high desire to carry carbon dioxide than hemoglobin bound to oxygen. This is the complete amazing story of hemoglobin and how it helps carrying oxygen and carbon dioxide. If you like this video, please subscribe to my channel and I will see you in the next one. Thank you.